And we are back. It is Monday morning, April 5th. We are finishing up chapter 27, Saccharides, Sugars, the sweet chapter. We'll get into 28 starting on Wednesday. I don't think we'll do all of it, but it's on lipids. We'll do a little bit on steroids. That'll probably be the new part there. We've already done quite a bit on fatty acids, but we'll do a little bit more there, show the synthesis of fatty acids. Homework 12 is due Wednesday then. Oh, but quiz 9, that's right, is due tonight, and that's the one that combines a little bit from 26, the pepsin hydrolysis using the 2 aspartyl side chains to protonate your peptide substrate and then bring in water to do the hydrolysis. So it is a peptidase there, I think the toughest one on quiz 9. But yeah, I think we're good there. And yeah, we only have another week of class. And we'll have time to review, of course, for the final. Now, don't forget the final after test 4, which will be next week. Test 4 starts next week. And then right after that, and I'll have to talk about this, it's by sections. So you have to come to a specific room depending on what section you're in for the final. And I'll need to talk about that. But the final is comprehensive. Remember, it's over 351 and 352. So be reviewing all the material. We have the sample final on Learning Suite already. The principles, again, I've tried to point out the principles of 351 all along the way here in 352. So hopefully they're not too distant in your memory, but you can keep track of those. Questions on anything? Okay. Let's go to the sideboard here and see if we can review a couple reactions. So draw alpha glucose. And alpha implies it's going to be in the pyranose form, in the ring form, right? But you should be able to draw the Fisher projection also, the right, left, right, right for the stereo centers. Let's just go ahead and draw alpha then for the sugar. So we got the equatorial, all equatorial there. And then it says to draw alpha at the anomeric position. So do we draw that next OH down, axial, or do we draw equatorial? Okay, most people were saying down. Okay, good. That seems to be consensus, and that's what it is, okay? The axial one, at equilibrium, that's actually the minor one. The major one is the beta, where they're all equatorial, but alpha means down in that position. Now, if we treat with methanol as a solvent with a little bit of acid, what are we going to form? We're going to form the methyl acetal. And notice I'm only drawing the methyl ether at the anomeric position, right? And I'm drawing alpha, and we could say also some beta is going to form, okay? We went through the mechanism of that last time. This is called a glycoside. It has an extra group on the side here. We'll see in disaccharides, polysaccharides today, that can be a sugar, okay? Or some other molecule. It can be an amine, like the blood sugar antigens that we'll see in a second. But yeah, just there, not these, okay? And that's because the anomeric position has that special resonance stabilized carbocation that we can get there. Okay, taking the same thing, alpha glucose here, and then treating with excess base and methyl iodide. Now, in the book, they also say the base can be silver oxide. Now, that can be kind of confusing. It is O2 minus, right? And that is basic, so it can be base. Of course, we know silver oxide is what the Tollens test reagent that does the oxidation to the gluconic acid. But here we're using silver oxide as a base, so don't be confused about that. I've mainly emphasized the base we're more familiar with, sodium hydride or sodium amide. Any strong base can do that. Taking this off if we have excess, and then excess methyl iodide, what do we get here? We'll get the methyl ethers at every spot. So every alcohol can become an ether at that point, including the anomeric one. Yeah, we can form the anion there and alkyd there. And let's see, methyl ethers all over the place. I'm drawing what, alpha? And so we should say beta is also there, okay? Because as soon as you put alpha glucose into solution, guess what? It'll start to isomerize to some of the beta, okay? So the equilibrium is always going on. If it's a free hemiacetal, okay? So don't forget to have the extra one there. Yes, question, please. Yeah, so person's asking, we pointed out in class that beta is the dominant one at equilibrium, the equatorial one. So is it better to show beta and then say plus alpha? Yeah, if you do that, you'll get full credit. I like to emphasize the alpha sometime because we're coming up here on the polysaccharides, and we'll see in starch that it's alpha linked. And I mentioned that that's usually the faster reacting one. It's called the kinetic product. Because it is the less stable one, but because of this anomeric effect being opposite the lone pair here and that sigma star overlap. So maybe mechanistically, that's why I tend to draw the alpha first. And alpha's down and then beta is equatorial. Yeah, but yeah, you can draw both and that'll be fine. All right, let's see. What else do we do? Oh, and then if we treat our all methyl ether here, alpha or beta, and treat it back in water and acid, okay? Here, we didn't have water, these first two steps. We just had some solvent, or methanol was the solvent. Here we got water, so what are we gonna get? We're gonna get the, back to the OH anomer, back to the hemiacetal. But look, all these other ethers stay put at that point, okay? So all of those are still ethers. It's just the anomeric position that gets hydrolyzed under those conditions. Okay. Here's another question just about structure. Can you draw alpha mannose? Let's see. Mannose is the same as glucose, except what? And it's alpha, so we should have that one down. And it is the D sugar, so we can add in those parts there. Alpha and the D means this guy's up in the back right here. And then what? We have equatorial, equatorial. Well, what's the difference one now at the 2 position for mannose? What do I need to do? Not the equatorial one in this case. Remember, it's epimeric or isomeric at that 2 position. So what do I need right there? Not equatorial, but axial position. Okay. And yeah, that crosses over a bond there, so you should be kind of clear there. That's coming out there. You can always draw in the rest of the hydrogens. See, at the 2 position, you've got the OH group there going to the left, you could say, from the Fisher projection, and that will be that position epimeric. How about galactose? Same thing as glucose. Oh, we're doing beta. There's beta finally. Okay. And it is a D sugar. So we got that there, and everything's equatorial, just like glucose, except at what now? The 4 position. So if I drew this equatorial, that would be glucose again. But to draw galactose, what do I need to do here? This needs to be up, okay? Some people are pointing up there, and yeah, that needs to be axial, okay? That 4 position, okay? So that's the difference between glucose and those other common sugars variations there. All right, a couple other questions here I think we can handle now. Here's ribose. We can ask you what kind of a sugar it is based on the generic name. It is a aldo what? Pentose. Okay. How many stereocenters? 1, 2, 3 now, not 4 in the Fisher projection. But we're going to draw the alpha furanose form now. Oh, furan refers to not a 6-membered ring. These are all pyranoses, okay? 6-membered ring. Furanose means what size ring? Not 6, but 5 now, right? And so, you know, and this kind of relates to what you've seen about DNA already, but you can tip this on the side, right? And you can see these 2 guys are to the right. And so when you tip it on its side, they're down, okay? And remember, this front edge is coming out at you, okay? And then the last stereocenter, it's still D, okay? It's to the right, which means this position here is up, okay? That's still R. If you want to do the CIP rules, assign R and S there. And then which one are we drawing? We're drawing alpha. So is that up or down? And that's right here. we're going to have to draw it what? Down. Okay. That would be the alpha furanose form of what? Ribose. Okay. Questions on that? How to come up with all that? Okay. So yeah, the principles here relate a lot to the naming, I think, but it has fundamental connections to the structure. Okay. And keeping track of that can be a little tricky with sugars, right? All right, now here we have a glycoside. We have something at the anomeric position. It is a full acetal. We can recognize that. I think most of the things we've had up here, yeah, these all starting out are what? Hemiacetals. No, these are acetals right here and here. And then, yeah, those are all hemiacetals there. But this one here starts out with some sort of ether there, some sort of R group. And that can vary. It can be a simple, methyl or ethyl group, or it can be another sugar. And we say that this carbon here has an ether there and an ether here. So that's indeed a full acetal. And we can hydrolyze that back. And the question here is supply a mechanism. What do we need to do first? Well, we're going to get to this product, right? We're going to get to the hemiacetal. So how do we get this off here, this ether off, and put water on there? This is a hydrolysis process. We could have an enzyme do this, right? These are called glucosidases, okay, or amylases, enzymes that at neutral pH and very quickly in water can hydrolyze. They're hydrolysis reactions using water to break a bond. So what can we do here? Well, we've got acid there. Let's go ahead and protonate the anomeric ether oxygen there. Or if we have an enzyme, you see we'd have some sort of a protonated basic side chain that we'd protonate, right? So a glucosidase or an amylase would work like that. So once we protonate, what would that look like then? Well, we get the protonated form here. Everything else on there, I'm not going to retry it. But you can see what that intermediate looks like, right? It's the protonated acetal. Let's go ahead and use a pair of electrons to come down and knock off that group. So our intermediate would look like that. And, you can draw either resonance form here, but notice by breaking that bond, we get the resonance stabilized carbocation, right? And we've got the sugar that was on the side, or the alcohol, or whatever our group was, coming off there. And then what do we need to do with water? Oh, and then just attack here, right? And then, to finish it, we've got to take the proton off with water, okay? So the mechanisms are similar. Hopefully you're comfortable enough with that. Here's a bonus one. Let's see. Number 69 on the homework. Molecule called fucose. And we're going to see fucose here in a second with the blood sugar antigen, guys. And fucose has this pattern. And yeah, if you look at the Fisher projection, it looks like that. What's different here? And the question asks you, what are the two main things that are different now or unusual about fucose? Well, it is a sugar. It's a hydroxylated aldehyde. Stereochemistry, what could we say here? Left, right, right. Oh, left. So is it a D or an L sugar? It's L, okay? Because this last stereo center is to the left, okay? If you assign priorities here, it's S, okay? What else is unusual here? Let's see, is it a aldo? Yep, aldose. 1, 2, 3, 4, 5, 6. Yeah, it's a hexose. But what else is unusual here? Who can see it? The 6th position, right? What's unusual there? Yeah, it's just a methyl group. Normally we had another alcohol down there. So we say this is reduced. It's a reduced sugar. It's a deoxy sugar. And we'll see deoxyribose in where? DNA. Okay, that's what the name deoxyribose means. Here in fucose, it's deoxy 6 position. Okay. Now, when we cyclize this, what are we going to do here? And I don't think the question asks you about this, but let's draw alpha fucose here. So we'd have this, that, I think up here, down here, and down here. Oh, and look, it is L. So look, this stereocenter right here is S, and I'm drawing it down. And I'm keeping that same convention for how we've oriented the sugars before, right? So here is the flat Hayworth projection, it's called, with the pyran flat there. But the oxygen's in the upper right corner. So look, this stereocenter, we've already drawn it up if it's D, right? But look, fucose is down right there, okay? And then you see the left, right here, left, right, with the up, down, down. So that's a connection. Use your models if you need to. Go through it, you know, a couple times at least. That's the way to really lock it in. And you can bring your models to test 4, you know, if this is a challenging issue for you. Let's see, let's go ahead and do the chair form then. If we put it in a chair, we'll have that one down over there. We'll have this OH down there. What about this one here? That will be what, up? This will be down. This will be, okay, that's one ring flip of it. And again, you see the down position here being the L sugar, and I think we got alpha here, yeah, because this is down. There we got the up one right there, down, down, yeah, and that's down. Axial, equatorial, remember they alternate. Might be better to do the ring flip version of this, right? If we ring flip this, then what would we have? We'd have this there. We'd have OH in the back there. We'd have this one equatorial, this one equatorial, and what? This one also equatorial. That might be the more stable form because there's more of them in equatorial positions. This is a bad thing with the axial here, axial here, axial here. You've got 3. Oh, and another one there. And I don't think problem 69 makes you go through that. They just ask you about the unusual features there. But I'd like to show a little more detail of how to analyze these harder problems. This is a challenging problem, right? We've thrown a couple of differences at you, right? The D position here, and then analyzing the different ring flip chair forms here. And the alpha, we can still assign alpha being down there. Okay, questions on any of that? All right, let's get into the disaccharides, polysaccharides. And go up here just for a minute, Jeshurun. Here we're seeing on the overhead a couple of di sugars. This one's alpha linked here. And we'll see that this will come from starch, okay? This will be maltose. And we see this one's beta linked between 2 glucose. So kind of hard to see whether it's glucose or not. But you see the all equatorial thing, the up position in the back there. If these are spinning and rotating, you'd see these a little better. But let's see where these come from. This one on the left comes from what? Starch, because it's alpha linked between the 2 sugars. This one will come from cellulose, because it's beta linked. You can at least see that or appreciate that from those models. But let's see what we need to talk about here. Let's come back over to the sideboard and let's take starch and we'll review that structure. We've shown it to you, and we're going to just partially hydrolyze it with some acid and water. You have to heat this up, and we're only going to get out a minor amount of product here called maltose. We get some glucose if it breaks all the way down, or we get oligosaccharides that are, you know, much longer than just the 2. So we're looking at disaccharides right now. So the disaccharide is just going to have 2 glucose molecules stuck together because this is polyglucose. And like I said, what's the linkage there? Well, it's 1,4, and coming off the anomeric position, what? It's alpha. Okay. And that's kind of the key thing here. And the alpha linkage is going to be seen in the disaccharide maltose. So let's see what it looks like. And we've got the alpha link. And we've got the other sugar here. And let's see, let's add everything in. And yeah, this is where these things get really complicated. But you need to focus in on the key aspects of that. That is glucose, right? You can see everything's equatorial there, except the alpha linkage here at the 1 position. So this is a glucoside. On the side here, we don't have a simple alcohol now. We have another sugar, and it is indeed what? Glucose. And it's linked to the 4 position. That stereocenter is set, right? That's the left, right, right, right. Okay? Right, left, right, right. That's the right position there at that 4 position. That's linked there, equatorial, equatorial. And let's see. Oh, we're trying alpha maltose because that alpha linkage is on the free position there. Now we've got a couple different acetals here, right? We have a hemiacetal here, and we have a full acetal here. So when we form maltose from starch, notice it can equilibrate as the alpha and plus what? Beta right here. Now that's the complicated thing. But this acetal position came from, what did we say, starch? So this remains alpha. This is not equilibrating under these conditions. That stays put. So you see the down position here. You can see down, I've drawn alpha, which is probably the minor one. The beta one's probably the major one. Question, does it mutarotate? When you isolate maltose and put it in a polarimeter, you can purify out the pure alpha or the pure beta, and that just refers to this position here. And what? Will it mutarotate? Yes, because it is a hemiacetal right there. So the optic rotation will change over time from pure alpha or beta here. Okay, this stays intact, okay? That linkage came from starch. Another question here, Tollin's test. Positive or negative? Is there a free aldehyde present? Well, not in our drawing here, but notice we are in equilibrium with what? With the open chain form here, where we could have this aldehyde that would get oxidized, right? Okay, so that is a minor amount. Why? Because it is a hemiacetal. So the Tollin's test will be what? Positive for maltose. Okay. All right, questions on that? Very similar analysis here on cellulobiose, which will come from cellulose. Okay, let's take cellulose and do the same thing. We've got to treat with acid. We have to heat it up a little bit. Cellulose is much more stable, actually, than starch. But if we do that, we'll get out cellulobiose. And it's also a disaccharide just of glucose, okay? And at the anomeric position, we're going to have another sugar stuck on there. And you know, this is a small product, you could say. There's a lot of other byproducts here, but if we get out enough disaccharide, it'll look like this. Well, wait a minute, that's just all glucose again. What's the difference? Is this the same as maltose? Well, same in one way. They're 2 glucose stuck together. What's the difference, though? What do you see right away? Cellulose is what? Beta linked. So this linkage right here at the 1 position coming off here is the beta. Okay, it's equatorial. And it's also 1,4 linked. And you see this is still, you know, this equatorial position off the glucoside on there. So it's really the same. It's 1,4 linked, but it's beta 1,4 linked now, okay? So when we partially hydrolyze cellulose, we get out this disaccharide called cellulobiose. Let's see, let's ask the questions here. Does it mutarotate? Because it is an acetal here, and it's still a hemiacetal here. I'm drawing the alpha cellulobiose right now, but we should recognize we also have the beta form, which does not refer to this, although the linkage is beta in this case, right? But this analysis over here, we're referring to the hemiacetal, which means what? It does mutarotate if we purify alpha or beta cellulobiose. And what about the Tollin's test? Positive or negative? Yeah, it's got a hemiacetal, so it's going to be what? Positive. Is that always the case for polysaccharide? And are they always 1,4 linked? And are they always glucose-based? No. We can vary this now. How about lactose? Let's see if we can do lactose here. So we're going to look at this disaccharide. It comes from milk sugar. It's in a variety of places in nature, lactose. And let me draw the structure here, and then you're going to figure out what the connection is and what the sugars are that are involved. So let's draw out lactose and see. These sugars always take a while to draw, don't they? And let's see. Oh, let's stick with alpha. So we're drawing alpha lactose here. Okay. It is a disaccharide. What's the linkage? Let's see. Well, here's our anomeric position. That's the 1 position, right? Is it alpha? No. It must be, I drew equatorial, so it's beta. Okay. Let's see. It goes over to the 4 position here. Yeah, equatorial, equatorial, equatorial. And then there's your alpha position on the hemiacetal. But notice the acetal linkage between the 2 sugars is indeed beta. Okay. It's also 1,4. Wait a minute. We just did beta 1,4 linked disaccharide with Cellulose. Is Cellulose the same as lactose? What's the difference here? We need to look at everything, right? Equatorial, 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 equatorial. That's indeed glucose. Okay. What about this one? Equatorial, equatorial. Oh, did I draw that right? Yeah, that's coming up. That's parallel to the axis. The equatorial position's at hydrogen. So what sugar is this? It's not glucose anymore. What is it? It's one you need to know. Yes. Galactose. Yeah. So this is Gal 1,4 linked to glucose with the beta position. Okay. Now to use this sugar in the diet, you need an enzyme called lactase. Okay. And people can be lactose intolerant if they don't produce enough of this enzyme. And that can be distressing to the GI tract sometime, because that'll leave a lot of carbohydrate available to the bacteria in the GI tract to grow on. But if you can produce enough lactase, you hydrolyze it to galactose plus glucose. Okay. And you no longer have lactose, milk sugar. You have the 2 monosaccharides. Okay. And you can imagine what lactase has. It has a acidic side chain that can protonate here and allow this to hydrolyze, and then water can be brought in. Lactase is indeed a hydrolase, okay, that hydrolyzes that and brings that in and generates the 2 monosaccharide. Questions on lactose? Okay, so very subtle difference, right? Just that simple difference here between this position here. And by the way, you need this galactose there. If you have 2 glucoses stuck together, Cellulose, that won't be recognized by this enzyme lactase. It only recognizes this substrate, milk sugar, lactose, okay, with the correct stereo center right there. Anyway, okay, let's get into, what's our next one? Oh, sucrose, table sugar. Oh, by the way, did we ask about mutarotation and everything here? Yeah, this is hemiacetal, so it does mutarotate and it is positive tollens. Is that always the case? Well, I don't know. Let's see. We got something else here. Yeah, I think we do. Let's look at this sugar, sucrose. And we're going to give you the designation here for sucrose, table sugar. Comes from sugar cane, comes from sugar beets. A lot of other plants have it. Those are the most abundant ones that produce the commercial table sugar. This is the stuff you bake with and cook with and everything. But what is it linked? It's 1 alpha glucosyl beta fructoside. So what is it? Beta 2. I needed a 2 in there, sorry. Oops. 2, there we go. 1 glucose, and we've seen that, glucose linked at the 1 position. Fructose, we need to review what that is, right? Fructose is the ketose, and it has the left, right part similar to glucose, but right, this is going to form a furan linkage, right? And we went through that last time, and the linkage ends up looking like this. And what we're going to have the beta linked position here. So our last stereocenter is there. We go around here, we're going to have up, down, and then we're going to have like that. Okay, and that's this front edge coming out at you, right? And you see at the 2 position, we have our ketone. And then if we turn that on the side, you see we got up, down, and that's that position right here. And it's still a D sugar at that 5 position right there. Now we're going to reorient this. We're going to rotate this around, I think, just 180 degrees here to get this orientation. And this will let us form, I think, the linkage here to glucose a little better. Okay, so this is just rotated around, right? You can still see this down right here, this up right there as it came around. And we've got this oxygen in the front now. Okay, and then you see the up down thing, which still has that linkage. Okay, now let's put the glucose on here and see if we can get the right orientation here. Yeah, and I'm actually drawing glucose a little bit different too, but you see all the equatorial positions here. And then we got alpha linked here. So that's the alpha at the 1 position of glucose. And we've got that down, and then we're going to link it to this position here. And see, that's the 2 position here. And that's why I've rotated it around. You can tumble these around in space all you want, but that just keeps the orientation a little bit more direct there. And then we've got the furanose, the 5-membered ring. And that's indeed the 2 position there. Okay. And then up, yep, and down back there. And then up for that position. And that's still your D sugar. That's the 5 position there. Okay. So yeah, this is indeed sucrose. 1, 2 linked. Okay. And this 2 position coming off, like I said, is beta. Because it's together, it's on the same side of the ring as the last stereocenter, the methylene hydroxyl. And yeah, this one's a little complicated, isn't it? But this is the sweet stuff. This is sucrose, table sugar. Let's see though, does it mutarotate? What do you say? First of all, the linkage here, right? This linkage here is an acetal, right? What about this linkage here? There's an ether, and there's another ether. What about at this anomeric position? Is that a hemiacetal, or is it a full acetal? You could say, well, here's an alcohol coming off. You can see that alcohol is not directly on this carbon, okay? To be a hemiacetal, it has to be an alcohol ether right on the same carbon. This is 1 carbon removed. This is the 1 position here. So what is this linked? Ether, ether. So what is this linkage here on fructose? It's also what? An acetal. It stays in place. You have to use the enzyme sucrase, and you can do that. This enzyme's known. Sucrase, okay, water, pH 7. It goes to glucose and fructose. Okay. But if you don't have that enzyme in there, or don't hydrolyze this full acetal, it stays in place. So does it mutarotate? No. Non-mutarotating, because it's anomeric carbon linked to anomeric carbon. All the other saccharides, disaccharides we looked at before, is one anomeric linkage, but then there's another alcohol from a non-anomeric position. So this one stays in place unless we do something drastic to it. What about Tollens test? Will sucrose give a positive Tollens? Will it make the silver mirror? No. Okay, because there's no hemiacetal. And that's kind of the bottom line here in this structure for sucrose. Okay, it's disaccharide with no hemiacetal. Okay, so that's a unique one. Okay, you could imagine other sugars where we have the anomeric positions linked together, and they're known. They're quite rare, but yeah, this is the important one to keep straight. Questions on that so far? All right, let's take one of these disaccharides. How about the lactose? Let's take this and not react it with lactase. Let's react lactose with sodium hydride and methyl iodide, excess, okay? And then two, let's hydrolyze this with acid and water. And let's heat this up. Let's really hydrolyze this thing. So what are we going to do? We're going to put methyl ethers everywhere except at the anomeric position, okay? So what are we going to get out? We'll get out galactose, and I'm drawing alpha here, but we'll also get beta. But we'll have what? Methyl ether there, and make sure you draw that one axial. That has the methyl ether axial position, and then this one also is methyl ether. Okay, but there's no methyl ether there. Why? Because that was an acetal when we did the alkylation, right? So when we hydrolyzed it, now it's a free alcohol. Well, what about the glucose? Well, it's also anomeric at this position. We would have put a methyl ether on there to begin with, but look, it's the anomeric position. So under those conditions, it will come off. We'll have plus the beta. So yeah, keep drawing alpha, which is a minor one, but it's okay. They're both there once you hydrolyze it. But what is also on here on glucose? It's the methyl ether here, methyl ether here. Ah, what about at this 4 position that came out of lactose? What will it be? Will it get methylated during this first step? No, that's just an ether. But when it gets hydrolyzed, what does it become? It just becomes that alcohol right there, okay? And it is glucose, so it's equatorial, okay? And then, yeah, the methyl ether stays put there. Believe it or not, in the old days, this is how sugar chemists used to figure out linkages. So they could tell, oh, look, we have a free alcohol at this 4 position, free alcohol here. Maybe they were 1,4 linked like that. They weren't linked here at the 3 or the 2 position because those are methylated now, okay? But I think from these products, you can kind of see how they're linked up. These are all methylated here. So this must have been the position of the acetal on the left there. And maybe the linkage was right there. Anyway, some of these reactions can be used to figure out the connectivity. I think that's all we need to say there. All right. Let's see, polysaccharide synthesis. We'll get to that later. Let's go back up to the overhead here, Jesharon. Yeah, so there's our linkage there. And let's view this in full mode. Okay, so yeah, you can look at these 2 disaccharides, maltose, cellulobiose, and hopefully the alpha and the beta positions stand out there in analyzing that. How about this structure? Here we have a polysaccharide. We're taking off the OH groups. You can kind of tell how it's linked. You can kind of see the 1,4 linkage there. And you can also kind of see the equatorial position there coming off some of these rings. And what that's going to be, well, here you see an axial linkage right here. This one's a little bit twisted. You can kind of not really see that one. Right there, it's clearly axial right there too. This is a fiber of what then? Starch, okay? And here's another polysaccharide now with 4 linked together. But you see the linkage there. What is that? That's equatorial coming off there. That position's a little clear, but as it tumbles around or rotates around, It's a little bit unclear, but here you see the what? Equatorial. So this must have come from what? Not starch, but cellulose. Okay, more rigid thing. Now, in nature, there are different forms of starch. Amylopectin is a form where there's branching that goes on. It's not just all 1,4, but there are some positions that are 1,6, and another sugar can come off that position. So you can have a sugar at the 4 and the 6 position, and that's where this point is where you see a branch coming off. Now, in amylopectin, amylose itself is just all 1,4 linked. It's linear. But amylopectin has this branching. And it's like every 20th sugar has then this extra 1,6 link. Glycogen in higher animals, this is the storage form, the polymeric form of glucose inside of cells. These are large granules. You can actually see these under a microscope. And they have a lot more branching. It's every 6th or so. Glucose has a 1,6 branch. And that's so you have more ends here. So you can easily hydrolyze that off and make more monoglucose for energy needs. But in plants, it's less branched there. Let's look at the blood sugar antigens. These are on erythrocytes, blood cells. So the circulating cells, we talked about hemoglobin before. This is on the surface of an erythrocyte. That should be colored red. Sorry about that. There's a protein there with a surface amine, a primary amine. Which amino acid do you think that is? Primary amine side chain. There you go. You should be able to figure that out. One letter code is K. What's the word? Starts with an L. Lysine, very good. So this is a lysine glycoside, which doesn't have another sugar on the side right there. And it's glucosamine. At the 2 position, there's an amine that has an acid amide position on there. So there are other types of sugars. They're not all just alcohol hydroxylated. Here's aminoglucosamine. And it is glucose. You see it's all equatorial there, the position there. And then you get over to this one. What sugar is that? Oh, you see the axial position? So, I labeled it for you, galactose there. And what's the linkage? Oh, it's 1,4 linked. And is it alpha or beta? You can see that too, right? It's beta linked. So I'm showing you these examples from biology, but we're using the principles to go through and analyze things, right? And then this one here, fucose, that's off the homework. It has the reduced form of carbon at the 6th position. It's just a methyl. It's deoxy, you can say, but all equatorial there. This is D-fucose. The problem in the homework was actually L-fucose. And then this one here, galactosamine. Okay, so this is galactose again with the axial position. And the amino that. Okay, this tetrasaccharide is in A-type people. How many people are A-type blood out there? If you know your blood. Okay, someone does. Okay, and then B-type, same thing here. What's the difference though? Glucosamine. This is just galactose without the amine there. The only difference here is this acid amide nitrogen you see is back to just regular galactose. But all the linkages are the same there. That's B-type blood. Some of you are probably that. Some people are AB. They have both antigens on the surface. A lot of people are O-type blood, which is just a trisaccharide. You see, if you only see 3 sugars there, you call that a trisaccharide. Same fucose, you can see the reduced guy there, galactose, and then glucosamine. Now, I'm not getting into all the immunology here, but if you have one type of blood here, then you have antibodies. Your immune system recognizes other types of blood as foreign, and it'll actually mount an immune response against that. Yeah, and there's some issues here. A-type people are universal donors because people who have AB type blood, you can actually lose this galactose easily here. You see it's alpha linked by the anomeric effect. This can pop off easily, and that if that comes off, it goes to the trisaccharide. But yeah, don't worry about that, okay? And what type of blood you can receive and all that, don't worry about that. Okay, just markers in the immune system. Now, the last couple topics here, and we'll do DNA on Wednesday. We'll probably not have enough time to. I want to do a little rational drug design. I've shown you Tamiflu here a couple times. This is the antiviral drug that's been developed here the last 20 years or so. we pointed out some of the synthesis here of what it does. But here's how it works, okay? They actually did the clinical trials with a 1-800 number. I got flu. They'd send you the drug within 48 hours, and you had to report your results there. If you take the drug within 48 hours of getting infected with influenza, 90% of people have it go away, the side effects. So it is very effective. And here's the basic biology of it. On endothelial cells, brachial tube cells, these are lining the air passages in the body. This is your cell, okay, higher animals here. It's a glucoprotein that's membrane bound, and it has this sialic acid on the surface. So it's a disaccharide, which is quite complex. This is a 9-carbon sugar, okay, which has an anomeric position at the 2 position, and it is an acid, carboxylic acid. It's oxidized at the 1 position. Surface of the enzyme of the virus has an enzyme called sialidase. What do you think sialidase does? It hydrolyzes sialic acid, okay? And by doing that, you can protonate here and bring in water. It's a hydrolase, okay? That simplifies the sugar on the surface of your endothelial cells and allows the virus to merge with those cells and then infect your cell, okay? So it has to simplify the sugar in order to merge with the membrane. And that's kind of a steric effect, you'd say. These saccharides are blocking the merger initially of the virus with the cell. The enzyme hydrolyzes it, transfers the proton, and now you can kind of look at the structure of Tamiflu and say, okay, here's the flattened resonance stabilized carbocation here. Here's the 6-member ring. And look, there's a 6-member ring with an alkene right there in Tamiflu. And that's where they started to design this drug. This drug is completely synthetic. It's designed just from the knowledge of the crystal structure of the sialidase bound to the sialic acid. Okay? They went from that point to design the drug. And you can see here, they made hundreds of different variations here. Here's the assays, the testing of the drugs with the sialidase enzyme. The lower this number, nanomolar-wise, the more potent it is. You see the actual drug here is potent at 1 nanomolar concentration, extremely potent. Binds more tightly than the natural substrate. And then the assay with the actual virus, okay, these are the natural viruses. It inhibits those also at low nanomolar concentration. So this is the one that became the drug. You see some others weren't as effective. Really, the breakthrough was to put this 3-pentyl nonpolar group right there. There's a pocket that's in the enzyme where that's located here, which is nonpolar. You can kind of see. So the crystal structure is known for the sialidase with the drug in purple there, bound to the interior of that. So that gives medicinal chemists, biologists, biochemists a lot of information to work on to change the structure and develop that drug. Okay, speaking of the immune system, we have the innate immune system, which is a bunch of enzymes that can hydrolyze different molecules in different pathogens as they might infect you, either on the skin or somewhere inside the body. Lysozyme is the most well-known one. It hydrolyzes the peptidoglycan cell wall of bacteria. So here's the peptidoglycan, which has some polycarbohydrate associated with it. Remember, they're complicated, those bacterial cell walls. And this enzyme right here, what can hydrolyze this anomeric position, right? This is the acetal. This enzyme can protonate that and bring in water, hydrolyze that, and that compromises the cell wall. The steps are known here for the mechanism. It involves some aspartic groups that can what? Protonate that position, bring that off, okay? And then leads to the hydrolysis, leads to the 2 sugars coming off there. And water has to go on there. And, we're leaving out the protonation step. But anyway, those principles carry over. Now, for next time, we'll go through the rest of the story here of the sugar deoxyribonucleic acids. And we won't have you memorize the structures of all the bases, okay? But you'll at least appreciate and be able to look at these nucleosides, as they're called, with the bases, what these are nitrogen glycosides, right? There's something different on the side here, not an OH group. It's not a hemiacetal anymore. It's a full acetal, but with another heteroatom, a nitrogen. So here's thymidine, cytidine, adenosine, and guanosine. You've seen those before, right? A, T, G, and C. You've probably just memorized the letters before. But they're deoxy at the 2 position. This is in DNA. RNA will have an OH group there. down from ribose, right? And yeah, we'll get into that next time. We'll have a little discussion on the double helix story, how it was discovered. The base pairing, hydrogen bonding, you've probably heard about before. Structurally, you'll need to know that or recognize that. And yeah, we'll get into the double helix, the hydrogen bonding between the base pairs, the complementary sequence. We actually do replication. And what's the other thing we'll do? Oh, more drugs. We'll do translation also, okay? And that'll be it for 27 here. So be reading ahead into 28. We'll start that also on Wednesday. Questions on anything? Come on down if you got questions. Yes, please. What's that again? We won't do. I'll be as clear as I can on that on Wednesday. Like I said, we'll do some fatty acids and we'll do some steroids. Maybe a little terpenes too. There's a couple topics in there, but they all relate to reactions we've already covered before. So question was on 28. What are we covering? What are we not covering? I'll be more clear on that on Wednesday, hopefully. Okay, very good, guys. Thank you.